Welcome to this episode of Eureka, and we are at Calcutta. We are at one of the oldest scientific institution in India, Indian Statistical Institute. This was founded in 1931 and today it is headed by Dr. Sanghamitra Bandyopadhyay. She will be our guest, and before we continue this conversation, let's take a quick look at her brief profile and we will carry on this conversation after this profile.
Computational biology and bioinformatics is an interdisciplinary field that develops and applies computational methods to analyze large collections of biological data such as genetic sequences, cell populations, or protein samples to make new predictions or discover new biology. It is where Dr. Sanghamitra Bandyopadhyay has extensively worked. Currently, Dr. Sanghamitra Bandyopadhyay is the director of the Indian Statistical Institute in Kolkata. She joined this institute in the Machine Intelligence Unit as a faculty member in 1999, after completing her PhD from the same institute. Her areas of research interest include computational biology and bioinformatics, soft and evolutionary computation, pattern recognition and data mining. In these areas, she has published more than 300 research articles in various journals, conferences and edited volumes. Dr. Sanghamitra Bandyopadhyay has identified a genetic marker for breast cancer, determined the co-occurrence of HIV and cancers, and helped understand the significance of the brain's white matter in Alzheimer's disease. She has published, authored and edited 6 books from publishers like Springer, World Scientific and Wiley. Dr. Sanghamitra has received several awards and fellowships, including the Bhatnagar Prize, Infosys Award, TWAS Prize, DBT National Women Bioscientist Award, G.C. Bose Fellowship and Swarna Jayanti Fellowship. She is a Senior Associate of the ICTP and Fellow of INSA, INAE, NACI and IEEE. She is also currently a member of the Science, Technology and Innovation Advisory Council of the Prime Minister of India.
Thank you for being with us in the show. It's a very old institute with a long history, played very important role in India's development, of course, about which we'll discuss later. And first I want to ask you this question, which is very, very curious. I mean, I was looking at your, let's say, educational career. I found that you had done BSc Physics.
Yes.
And currently, if I look at everywhere, all over the world, whoever is citing, giving citation for your award, they are saying that you are working in the area of computational biology. Yeah. And for most of us, physics and biology are like chalk and cheese. I mean, how did you marry these two things, which for most of us looks like two opposite poles?
Right. So yes, I was doing my BSc in physics. And then I wanted to do something different, so there was an opportunity in Calcutta University to do a post BSc BTech, which I did in computer science, and I think that was good for me because I liked computer science very much. And then I went on to do research in computer science. I was doing optimization, pattern recognition, data mining, stuff like that. Then it so happened that a student met me and wanted to do his research project with me, and he had a biology background.
Okay.
He brought in this biology aspect into my work and convinced me that there was, at that time 2004-5, it was something new, a small microRNA called small RNA molecule called microRNA. Mm-hmm. People were getting excited about this molecule.
Okay.
And he convinced me that there's enough of computational work which needs to be done in that area, and that the methods that I have been working with for the last several years could very well be applied to this area. So in my school days I was terrified of biology. I was terrified of anything which I perceived required a lot of memorization. I was very bad at memorizing things. So that is why I took to computer science very nicely,
because it's more
it was
logical.
Logical,
Yeah.
right. You didn't have to memorize too many things. And then the student, he got me into biology, but since my biology knowledge was very weak, he taught me biology, I taught him computer science and mathematics.
So the student and teacher were teaching each other.
Certainly,
So,
I think that happens all the time.
Yeah.
It's never a one-way traffic,
Yeah.
especially in research. The student, of course, gains a lot from his or her interaction with the supervisor, but the supervisor also gains by interacting with the students. And that is why I think even though there's a perception that academics pays very little, research does not pay that high, but still there are many people, many like us, who are very much in love with research, because
it gives a personal
it's a
satisfaction.
constant interaction with the students which keeps us energetic, keeps us young.
So what happened to that student now?
So that student actually did a fantastic work. He went on to do his PhD also with me as a co-supervisor, and the qualities that he had, his academic background was not very good. In fact, it was very ordinary. But the qualities that he had was a very high level of sincerity and hard work. And he worked with me, and we published some very good papers. After he finished his PhD, he applied for a postdoc position in Stanford University. So given his list of, actually that was also recommended by another professor in Columbia University who had looked at our paper, and he found it so useful, he got in touch with us, and I put him in touch with the student who answered several of his queries. So that Columbia University professor had actually used our method and found it to be very useful, and he recommended to his colleague in Stanford. The Stanford University professor then asked for transcripts of the student. After getting the transcripts, this professor was very confused because the marks were very ordinary. So then he called me up because he was confused whether the student had actually done the good work or it was the supervisor's work which the student was just included. So he called me up, discussed with me about the student for about 45 minutes, and my main response was, if you want to take the student, take him. Do not think so much about the marks, because this person will be an asset to any organization that he goes to. And he was then offered, and he then that was several years back, and now he's moved on. He's got to another university, and he's got a very junior faculty position there, but he's doing very well, and he continues to work hard. And the feedback that he tells me is that many of his colleagues in the US, in the laboratory where he was a postdoc in different universities, they did not survive the hard work there. But he said that I survived because he acknowledges that he got the training of working very hard in my group. So that is something which I am very happy about, and the student has done brilliantly. His name is Sanghamitra, and he of course continues to be in touch, continues to collaborate, but that once again reiterates the very cliche statement that there is no alternative to hard work.
Most of your work which kind of had international recognition is also related to using computational methods for biological problems.
Certainly.
So now we are using two terms, computational methods and biological problem. So let's understand this word computational method.
Right.
So when you say computational method in simple terms, what are all it? I mean, it's only one or multiple?
Oh, multiple.
So can you say about one or two,
Right.
like illustrate so that we'll have an idea of what is this computational method
Sure.
is?
One thing I must say, that because I am essentially a computer scientist.
Yeah.
Being a computer scientist, I take pleasure in developing methods, computational algorithms, which will beat the existing methods, and at the same time will be good for biological problems, will give us better insights than existing methods. So my target is not to use existing methods for solving biological problems
But
that the
creating a new
biologists will
method.
do.
Yeah.
That the biologists would
Yeah,
do.
yeah, yeah.
Okay, my pleasure is in developing the methods,
Yeah, yeah.
methods that will beat the state of the art. So that is what we do. We have excellent methods in optimization. So optimization is very easy to understand. Suppose you want to go from one point to another point, you want to optimize, let's say, two things. You want to optimize the time and the cost, and immediately you can see that if you want to minimize the time, the cost will go up. You want to minimize the cost, the time will go up. So these two cannot be optimized at the same time. how do you tackle such problems?
So
there we develop methods.
out of the multiple methods, you find that this is much
Not
better.
multiple methods, we find
now
what is the
multiple
best.
paths. Multiple
yes,
paths.
that could be multiple paths, yes. But we develop the methods of tackling all these paths.
Okay.
And there are problems where, if you think a little, the number of paths is enormous, exponentially high. The term exponential is very important because that is where the complexity of the problem comes in. If it's multiple of something, then it's not too much of a problem. Let's say 10 times 1, 10 times 2, that is fine, but 10 times 10 is huge.
Yeah.
Okay. So 10 times 10, no, I'm sorry, 10 to the power 10.
Yeah.
When it goes into the power, 10 times 10 is nothing. 10 to the power 10 is huge. Okay. So that is what exponential growth is. So our methods are applicable where problems are really, really complex. Then that is one part.
So you said that a computational method, for example, optimization is, for example, one. Maybe another example of computation.
Yes, exactly. So suppose you have many objects. Okay. In computation, in our terms, we will say many points in a space. Okay. But let these be objects. It could be human beings, for example, which are represented by some features. Okay. Your height, weight, circumference of the head, etc. could be some features by which I represent you. Similarly, I represent millions of people. Okay. And that is all that I have. What do I do with that data? The first thing that anyone would try to do is try to find out if there are some patterns, if there are certain individuals or certain objects which are very similar to each other, group them. Okay. If we don't know anything else, then what we typically try to do is group them. Even if we have a multiple pen or pencils lying on the table, what do we usually try to do? We usually try to group them
all the black pens together, all red pens together.
That's right. So that is natural. That is called,
or otherwise a long pencil.
Exactly. That is called clustering. Okay. So we try to cluster the data. Okay. And immediately you have given the answer yourself. There is not a unique way of clustering the data. You can cluster it in terms of colors. You can cluster it in terms of their sizes, maybe in terms of their thickness. So problems become different when the data set changes, but there is a lot of application, lot of problems in biology which require clustering.
This is where we will take a very short break. Keep watching Eureka. After the break, we are going to talk about what are all the kinds of biological problems, for example, computational biology deals. We will take a very short break. After the break, we will continue this conversation.
Tales that inspire. Stories of social change. A salute to diversity. Promoting public discourse. Events that motivate. Inspiring the innovative spirit. Watch Rajya Sabha television documentaries.
Welcome back to Eureka. We are having a very wonderful, very insightful conversation with Dr. Sanghamitra Bandyopadhyay. She is the Director of Indian Statistical Institute in Calcutta. Before the break, we tried to understand in a very simple way what is computational methods. I mean, one or two we saw. Now, let's take a quick look at what is biological problems which require this computational method. One or two examples, for example.
Right. I mean, it's a large number of examples, first of all. Biology, for that matter, anything else, cannot do without computer science, without statistics, without mathematics anymore. For example, the technology has so improved that you get huge amounts of data, and therefore you just manually cannot make sense of the data. So you take any problem. For example, you have genes. Okay, in the we know that the chromosomes inside the cells, we have the nucleus. Inside the nucleus, we have the DNA, and the parts of the DNA are the genes. Okay. So genes, we need to understand which genes, because genes will produce their product. Okay, what is the direct product of the gene? Usually, typically, a direct product is the RNA. And some of these RNAs, which are called messenger RNAs, goes on and produces the protein. Okay. So now you would like to understand which genes, for example, are involved in diseases. certain diseases.
Or
Take any
triggering a
disease.
like,
Take any disease, okay? You may take a cancer, for example. So, you take a set of patients who are clinically known to be suffering from a certain type of cancer and who are clinically known not to be suffering from that, okay? And then you look at the expressions, that is how much of the product a gene has made. Let's say the RNA.
There I want to ask a question, because many of us get this. For example, if you take a gene from me and gene from you, it will also have a difference, right? So, it is not like, for example, the patients will have one gene and the non-patients will have, I mean,
no, genes typically would be very similar. I mean, but if you look at the DNA, the genome, the set of genes, the entire DNA of the individual, there will be minor differences between you and me. But there will be, of course, there will be, but
that's
very minor
differences.
What makes us different. But the difference between you and me would be much smaller than the difference between, let's say, the genome of a chimpanzee, also chimp also, and a human being, or a chicken and a human being. So, these are all different, but your and mine differences will also be there, but will be very small.
So now we have two populations.
So, we look at the same gene of the individuals, of the different, and how much of the product the gene is making. Okay. So, under two different conditions, you are suffering from the disease, I am not suffering from the disease, or vice versa. I don't want you to put you in that troubled spot, but X and Y. Okay. So, so we take a set of patients and a set of normal individuals, let's call them non-diseased individuals, and we look at the set of human beings typically have of the order of 30,000 genes. We look at the 30,000 genes of these individuals, let's say 30 from one group, 30 from another group, so 30 patients and 30 normal. We look at the 30,000 genes and the expression values of these genes, how much of the product it has made. Okay. Now, you can think of like a table, right? You have 30,000 rows and you have 30 and 30, 60 columns. Okay. And each value gives you the amount of product made that each of them by that gene in that particular person. And that particular person may be a normal, non-diseased person or a diseased person. Now, you see, from this data you are trying to understand which genes may be responsible for that particular disease. Most of the genes would have no role to play. They would not show a remarkable difference in their expressions, in the amounts of product that they have produced between these two groups. Okay, most of them. So, you have to eliminate those, and you have to focus your attention on those where you see that maybe in the diseased samples the expression is very high, but in the normal it is seen that it is very low. So, that means otherwise. So, there you have to look for patterns which stand out in between these two groups, and then focus your attention on these this set of genes where the pattern is remarkably different. Now, you see, I am talking in very uncertain terms, remarkably different. You have to really quantify it, and there are different ways of doing this, and that is where the computer algorithms, the statistical methods, very important. Statistical methods will be very important here. The mathematical models, they come into the picture. How to identify the set of genes which are responsible for this disease, and it could be just an erroneous observation. It could be by chance that this has just been observed in this set. So, how do you build models so that your confidence level is high?
I think if I have to expand this, actually one of your work is actually on breast cancer, right?
Yes.
So, can you elaborate on that? I mean, because I think there is a link between the kind of way you are explaining your biological problem and the breast cancer issue.
Right. So, what we did was actually breast cancer and colon cancer. We looked at the data sets which are available. Please understand that we as computer scientists, we have not been generating data. We take data generated by others. Okay, and then we give the methods. So, what we did was we looked at this data of microRNA. So, expression, how much of microRNA is produced, how much of other RNA, the messenger RNA is produced, etc. We look at such data, and in fact, you see inside the cell, these small molecules are they are all inside the cell, they are interacting with each other, and these interactions you can model as a graph. Okay, graph meaning you have nodes and you have edges. The nodes mean entities, for example, the molecules here, and the edges mean two molecules would be connected if they are interacting. So, it is a very complex network which is there. Now, we do network analysis. We try to give methods for analyzing this network, which becomes a computational method. Okay? And from this analysis, we were able to identify a very small set of microRNAs in particular, and sometimes also other genes, other mRNAs, where we saw that they appeared to be playing important roles in many different directions, okay, because they were like a hub from where everything was like, it seemed like all action was taking place there. It was controlling many other things. So, and of these small set of molecules, we found that at least, let's say 5 molecules we were able to identify, of which 3, when we looked at the literature, we found that they were known to be involved in
So now you also have a confidence on your
on
method.
the remaining 2. No, 5 we have identified.
Yeah.
3 we have got evidence from the literature, gives us confidence on the method.
Yeah.
The remaining 2 could be important. People have not looked. Yeah. In fact, after we published, a couple of years after that, independently another group actually validated one of them. Independently. They were not looking at our method or whatever, but that was a vindication that what we had done was actually
So
also
it also
seen by
tells
others.
that if we actually apply this method, we will also know what
Hopefully.
kind of thing will be the more
Yes,
you know,
yes.
faster route to finding
Yes.
connections, right? Yeah.
Faster route, and there is no other way, but you have to use computational methods, because you see what I typically say is biologists are looking for a needle in a haystack, a very big haystack.
Exactly.
It's a huge amount where to look, where not to look, huge amount of possibilities. Now with computational methods, with these mathematical models, if we can reduce the size of the haystack, it still remains a haystack, because our computational method will not say, will not be 100% foolproof.
So
Okay.
it's something like, for example, digging a hole to find water. I mean, you can dig anywhere, everywhere, but you will not find water. But if you know at least
Some information you have that the
Yeah.
probability is high, that
Higher than
you
at
are
least,
likely to succeed here, you are very less likely to succeed
so
somewhere
then at
else.
least from here, I mean, the number of
That gives
effort
a
that you
handle.
put. Similarly, another work related to Alzheimer is also considered significant, because I suppose for first time it pointed out that the white matter also had a role and had an important role in Alzheimer's disease. Can you explain what exactly?
Yes, again we have been looking at this problem computationally. There was some data available for patients suffering from Alzheimer's. It was typically women suffering from Alzheimer's, and a set of patients who were not. And we were looking at microRNA expression data again, and we built a new method. We gave a new method there of switching patterns. Okay, it's sort of a graph theoretic method, like graphs.
Yeah,
okay, we were looking at differentially co-expressed switching patterns, which are slightly difficult to explain, but these patterns we found that they emerged quite well in the white matter. This was a surprising information, because so that indicated that there is some signal, important signal in the white matter also. Typically it was considered that the white matter does not have a major role to play in Alzheimer's, but it was also true that some very little bit of studies were emerging that maybe it is important to look there, but primarily people focused on the grey matter. But this sort of gave an indication, our results computationally, that there are important signals there, don't ignore, not to be ignored. So that is what was
that's
interesting.
a kind of a new insight to the way you look at it.
Yes.
This where, I mean, slightly I want to change and ask something about your journey. I believe it's a push that your mother gave that actually brought you to this place.
Yeah. So my mother has been a very big influence, and my grandmother also. But my mother, she never really brought me up like a girl, in the sense that of course I was brought up like a girl, no doubt. But the restrictions which are usually imposed, not much restriction, rather she would push me to do certain things which typically mothers would not. I remember one incident where I was visiting my uncle's place, and they had a big garden with a tree, and I wanted to climb that tree, but I was not getting enough confidence. She told me, go up, I am there standing below,
Okay,
if you
that's
fall I
nice.
will hold. But the point is, that was an important push, psychologically actually.
Yeah.
The other very important push that she gave me was during just before my master's, MTech in computer science. I completed my BTech and had a very good result in my GATE examination, which would enable me to study in MTech in IIT Kharagpur. And I think that was a very good move for me, because IIT Kharagpur actually molded me as a person. It really taught me the meaning of hard work, and I had great time there, worked very hard and enjoyed every bit of it. So I got my first grey hairs in IIT Kharagpur, but it was so enjoyable. And then of course I came on to ISI where I did my PhD.
This institution was formed in 1931 when the British was ruling India with a particular purpose. I mean, they wanted to collect statistics so that they can rule India in a better way, I mean, govern India in the better way so that they can earn their. But after independence, one of its important role has been in the national planning, so national planning. Today, what do you think is the key role for this institute in India's development?
Yes, first of all, I'll correct a little bit. This institution was formed by Mahalanobis because he himself realized that for a country for the human welfare and for national development, statistics is the key. So the data collection, data analysis, new methods, very solid, powerful methods, they are necessary. So that is how this institute grew. It was built by a small set of people, not really by the government, but by individuals who got together, who were dedicated to the cause of statistics. Mahalanobis himself was a physicist.
Yeah.
He learnt and he taught physics till he superannuated from Presidency College. And it is there in Presidency College that this institution was formed in a one-room laboratory in 1931, 17 December. Now, yes, as you correctly mentioned, it played a major role in planning of the country. Second 5-year plan was spearheaded by Mahalanobis. It did a lot of other contributions. Mahalanobis in particular led a lot of contributions in sample surveys, in sort of coming up with this Mahalanobis distance, right, and many other things. And it kept on, this tradition was kept alive by his successors, C.R. Rao, J.K. Ghosh. There are many, many names over there. And now also it has diversified quite a bit now. Statistics keeps on being the mainstay of this institution. Mathematics is very strong here. There's very strong economics programs in this institute, very strong computer science programs. In fact, the first MTech in computer science of the country was started in ISI.
That's interesting.
The first computer in India came to ISI, to this campus, to ISI Kolkata. The first computer indigenously built between Jadavpur University, in collaboration with Jadavpur University and ISI, it was called ISI JU, ECJU, here.
Yeah.
Okay, so that computers would be imperative to be used for data analysis was realized by Mahalanobis, by many other visionaries of that time. Bhabha also realized that. So, this institute, therefore, with Mahalanobis as his leaders, was thinking way ahead of times. And we continued that legacy. We have very strong departments, as I said, of all these areas. Besides that, we have geology, we have physics, we have statistical quality control and operations research is one of the very old divisions of the institute. We have many other social sciences, linguistics, psychology research unit, and many other activities which go on here. In this institute, we have a geology museum where you can find a fully mounted dinosaur fossil, excellently kept. So that is something we are very proud of. Anybody who visits this institute, that's a must see. We have Mahalanobis Museum, which tells the visitors about the contributions of Mahalanobis over the period of time he did his work. And even now we have been able to attract the best brains, the best intellects of the country, around the world. The topmost awards come to this institution on a regular basis. So our contributions are there in the government also. For example, our scientists have come up with mechanisms by which how much of fake currencies in circulation can be estimated. They have made contributions in Swachh Bharat. They have tied up with Tamil Nadu government, for example. We tie up with state governments to do projects that they want to be done by us because of our expertise. We collaborate with many other institutions. For example, we have a 6th institution collaboration going on now where we are looking at oral cancer and cervical cancer. So that is a DBT funded big project which 6 institutes in and around Kolkata, of which ISI is a part, which is looking at the data analytics part, the statistical methods and models, and also building the portal. So we are into various types of activities like this.
Thank you for being in the show. It's been a very wonderful opportunity. Keep watching Eureka. We'll come back with another episode next week, same time.